Hello, welcome to part 1 of the Unit 1 review for Bio 30. If you're getting ready for your Unit 1 exam, hopefully this will help to jog your memory. We talked about in this unit the brain and the sensory organs. Those are going to be in part 2. In part 1, we're going to talk about neurons and synapses. Neurons are one type of cell in our nervous system. We have lots more support cells or glial cells that don't send signals, but they're important for keeping the neurons healthy. So the neuron can be connected to other neurons, or it can be sensing the environment, a photoreceptor or a chemoreceptor or a mechanoreceptor or a proprioceptor. But those receptor parts are going to be at the ends here called dendrites. The cell body has things like a nucleus in it, controls the health of the overall cell. The axon is the long skinny part that we send the signal down. It can be really, really short or it can be pretty long. And sometimes the axon is wrapped in Schwann cells, which are a type of insulation that can help the signal to go not only faster, but also with a little bit less chance of short circuiting. These create something called the myelin sheath, which is just the sheathing or the insulation around that axon. The gaps in there, when you do have myelinated cells, there are gaps in between called the nodes of Ranvier. So the signal goes from here to there, to there, to there. It jumps. The jumping we call saltatory, from Latin for jumping, conduction. At the end, we have terminals, and the terminals are where the signal gets passed on to another neuron or to another effector cell. We call this an electrochemical process because the signal is sent with a change in, well, voltage. But the voltage is generated by moving around ions. So the electro part is talking about the charge, and the chemical part is talking about, well, we're actually moving mostly sodium ions and potassium ions back and forth. Nerves can be excitatory or inhibitory. Excitatory nerves are ones that are going to make it more likely for that signal to get sent on. There's always a threshold you need to pass before that signal can be sent on. So here A and B are both excitatory, but each one by itself is not enough. But if they work together, they can pass that threshold and start the depolarization part. Cells can also be inhibitory. You can have inhibitory neurons that make a dip, make it less likely for that signal to get passed on. Both have their uses, but for right now, we just need to notice what they look like and the effect that they have. When you're sending a signal down an axon, you are going from the rest state, where normally it's positive outside and negative inside. And for the duration of the signal, you've got a little bit of time where it's more positive outside and more negative outside, a little bit of a flip. So at rest, inside of your axon is going to be negatively charged, and the outside is going to be positively charged. That's thanks to the sodium-potassium pump. The sodium-potassium pump is this or this. It's a protein. And it's a protein that takes 3 sodiums, moves them inside the axon, and takes 2 potassiums and move them outside. And they're both ions, so they're both charged particles. They're all positively charged, which makes this not super efficient, but it does result, because we're moving 3 of them one way and only 2 of them another way, to a more positive charge outside and a more negative charge inside, which is what our axons at rest look like. When we send a signal, assuming we can get past the threshold, so if we don't reach the threshold, no signal gets sent, but if we do reach that threshold, then we open up those sodium gates, and suddenly these sodium ions that are waiting outside can rush in. And that is the signal. So while they rush in and things are heading towards 0, so 0 volts or 0 millivolts would be maybe around here, I don't know, and we go past that a little bit. This part is called depolarization because at least for the first part, we're heading towards 0. Once we reach our maximum, which is usually around 40, so plus 40 millivolts, we start winning the fight to put it back to normal, and we start doing repolarization, so heading back towards a polarized state, because at rest, it's polarized, right? It's got a negative bit inside and a positive bit outside. So repolarization happens in here, depolarization happens in here, and hyperpolarization happens At the end of repolarization, we overshoot our goal. Our goal is to go back to around -70 millivolts, but if we go a bit too far, that's okay and actually good, because hyperpolarization gives us two benefits. One, time to recover, called the refractory period, so that we don't have a constant signal from a nerve. It gives it us a chance to refresh itself. These pumps need a bit of time to get things back to normal. Another one is it prevents the signal from going the wrong way. Because the region just behind the signal is going to be a little bit extra polarized, it's going to make it even harder for that signal to head back the wrong direction. In our textbook, we say -55 millivolts, or in our notes, we say -55 millivolts is the number to remember because it's common, but it can change. And some people might have a lower threshold for certain nerve cells. So if you can smell really faint smells that other people cannot, or you can see dimmer light than maybe somebody else could, maybe that threshold might be lower. So that might be a lower threshold for you. The overall organization of our nervous system is broken into two main categories, central and peripheral nervous systems. The central nervous system is the brain and the spinal cord. Those can contain gray matter, and those ones don't have neurolemmas, which means that they can't heal if they're injured. So if you have a spine injury or a brain injury, this can be something that takes a really long time to heal up. In the peripheral nervous system, so all the other nerves all throughout your body, we have two divisions as well, the autonomic and the somatic nervous system. The somatic nervous system affects or is concentrated on things like sensation. What do I see? What do I hear? What do I feel? And of course, the business end, the motor end, the efferent end, where you are going to make an action to do something about those sensations. In the autonomic nervous system, we have two different divisions too, the sympathetic and the parasympathetic nervous system. The sympathetic nervous system we can think of as the fight or flight. If you are in danger, you might be more in need of, well, big wide open eyes or deep breaths or a fast beating heart. Or you might need lots of sugar, so the glycogen in your liver might be broken down into glucose. Or you might not want to pee right now. You might want to focus on just running away or fighting. For parasympathetic, sometimes people call this the rest and digest um, system. And in this one, just it's the opposite. So if you remember what's happening for sympathetic, okay, it's danger. For parasympathetic, you can just think, okay, the opposite things happen. I'm going to focus on relaxing. I'm going to focus on digesting my food. I don't need a lot of blood sugar right now. My heart doesn't need to beat really fast, that kind of thing. At the junction between two neurons, we have synapses, or at the junction between one neuron and something like a muscle cell. So if it's between two nerves, we call it a synapse. But if it's between a nerve and a muscle cell, we call it a neuromuscular junction, which is a cool phrase, but it's also a synapse. So at a neuromuscular junction, let's just take a look at this one. We have a signal that is going to reach that down here. It's a muscle cell. And that signal is going to be transmitted along the axon to get to the terminal. But when it gets to the terminal, that's the end of the cell, right? There's a wall there. We need to send the signal further on. So we have these little bubbles, these vesicles or vesicles, you want to call them, that are full of neurotransmitters. And those neurotransmitters are being made all the time. They're always being broken down and remade. And you've got lots of mitochondria around to help you do that and to help run the sodium potassium pumps too. So these neurotransmitters are sitting around waiting for a signal. When the signal reaches, they're going to come down to the end, and this bubble is going to break open. This is exocytosis. And those neurotransmitters are going to float across that synapse or neuromuscular junction. They're going to reach a receptor. Here's some receptors, here's a receptor, and those receptors are going to be keyed to just that one neurotransmitter. Once that receptor is activated, or rather once enough of them are activated, then that effector is going to do what it's going to do. Or if it's a synapse between two nerves, Then if enough of these are activated, then the signal is going to be translated into a new signal for a new neuron to send on. After that happens, the effector is going to do what it's going to do. So if it's a muscle cell, it's going to contract. But there needs to be a stop to this. Once these neurotransmitters are on the receptor, they're going to sit there forever, which is not great because otherwise you're going to get a signal that doesn't end. You're going to get a muscle that contracts forever. So they have to get broken down. The neurotransmitters are disposed of by an enzyme. In this case, it's cholinesterase to break down acetylcholine, cholinesterase. ASE is the enzyme ending. And that's the normal way of things. These parts from the broken down enzymes sometimes get reabsorbed, but at any rate, new neurotransmitters get created and this cycle can continue over and over. Sometimes that signal is not strong enough, and we can get around this problem by using an enzyme inhibitor. So how normally we want an enzyme to break these guys apart after they've done their job, maybe we want to slow that process down so that that signal can be stronger. So to do that, we just get an enzyme inhibitor, which is something that looks enough like the enzyme's target to bind to it, but not enough like it for get the enzyme to do its job and move on. So the enzyme gets stuck. This is inhibition. This is competitive inhibition, if you remember from Bio 20. So if we have something like that, say a cholinesterase inhibitor, then that signal will be amplified because these neurotransmitters will sit on the receptors for longer. If there was no enzyme at all present to break them down, then we would have a continuous signal. If your muscle got that signal and it never stopped, your muscle would be contracting for, well, not forever, you're going to die eventually, but for a painfully long time. So in general, we're going to talk more about sensation in video 2, but in general, we have some kind of sensory input that's going to go along an afferent nerve to an interneuron, and that's going to head to an efferent nerve to an effector. This is the shortest possible sequence of events that we could have. Every time that we say a neuron depolarizes, we mean it sends a signal, because once that depolarization wave starts, that's what a nerve signal is. If we look at a reflex arc, it's one neuron, the sensory or afferent neuron, the interneuron in your spine, and the effector or motor neuron, maybe your arm. If that's a reflex, your brain is not involved. Your brain can be involved, and in the brain, there could be greater than one neurons involved. There could be lots of neurons involved in the decision about what to do. But in a reflex arc, there's one to bring the signal to your spine. The spine passes the signal on to a motor neuron and says, okay, I'm in pain, I better move, or that's a fire, pull my hand away. In the second part, we're going to talk about the brain, the eye, and the ear. And my biggest advice for you in all of these videos is to do as many of the practice problems as possible. There's practice problems in your notes. Do as many of those as you can. It's going to be the best way to study. Ask questions too, if you've got them.